Thanks for coming. For those that don't know me, I'm Nathan Smith. I'm a master's student from Coastal Carolina University. And as everyone's kind of been mentioning, for the past couple years, I've been working on this summer catch and release mortality project with the striper here at Smith Mountain Lake. So just to kind of go over a brief overview of some of the previous research at Smith on catch and release mortality studies. They're mostly reviewed by Bettinger and Wild in 2013, and the main findings that that paper produced was that temperature and gear type were the main contributing factors to catch and release mortality. For catch and release mortality, typically that's going to be greatest during the summer, where in some cases that could be as high as 83%. But really the bulk of this work just contributed to pretty conservative management plans for striped bass across the country, and particularly in southeastern reservoirs. And the reason why they're doing this kind of work is because in some of these reservoirs, there's temperature-based striped bass fish kills. And so, there's questions related to, well, why is this happening? And why should we be concerned about it? And really what it comes down to, and what's been hypothesized, is that striped bass deal with something called oxythermal squeeze, where how that works is during the winter, when the water's the same temperature from the surface to the bottom, it's cool and well oxygenated and they can use that whole habitat space. But when we go into the late spring, as water's warming, the surface is warming way faster than the water at the bottom. And so what that does is creating a thick, warm layer.
You jumped one.
Yeah, I jumped like 3. Can you hit me in the back the other way? Yeah, thank you. But as that water's warming, it's warming too fast and it's making that thick layer of warm water at the surface. Well, once that water gets warmer than 77 degrees, striped bass stop using it and they avoid it, and so they sit a little bit deeper in the water. When we take that to the summer, the longer that spreads out, bacteria at the bottom of the lake end up using the oxygen that's down there and reducing it for everything else that's in the reservoir. So when you have that oxygen being depleted, by the time it's below 3 milligrams per liter, then striped bass are going to stop using that as well. So that phenomena you get is over time through the summer, striped bass are being restricted or squeezed into a narrow body of water, and they're not as able to use their resources as effectively. A little help. Keep going, a couple more. Yeah, here we go. So how does it look like with some reservoirs in our somewhat general area? Well, Lake Murray down here with that news slide was about, that's a reservoir that has striped bass that has been evaluated for catch and release mortality. And that 83% that I was talking about earlier, that's where that number comes from. And that's because that reservoir experiences a higher degree of oxythermal squeeze during the summer than some other reservoirs. Lake Gaston, it's on the same river as us. It's along the Roanoke River, and also it has some amount of squeeze that's restricted for striped bass habitat. Smith Mountain Lake's a little bit different. We don't have temperature-based fish kills here, and on top of that, their habitat is available for the entire duration of summer. And so what that kind of led to for Dan is like asking, well, how does catch and release mortality work here? And this is just an example of the water temperature data from the SML Plus app that all you guys probably have access to. And what's interesting is so this is the previous captain quarters, or known now as Marina 66, their temperature spots. And so the purple is going to be at 20-foot depth, the blue is going to be at 3 feet. And what we're seeing is over time, by the time you get to June, the 3-foot temperature and the 20-foot temperature just completely separate. And so that's defining that stratification period. That's when this catch and release mortality stuff is really coming into play. So if you're going and you're looking at SML Plus and it's, it's June or whatever, if you're seeing that, that's when you want to start thinking about some of the things we're going to be getting into. So why Smith Mountain Lake? It's a really awesome fishery. It's one of the best in Virginia, and the ability for striped bass to just get really big and be awesome for people to catch is kind of the main reason why people come here, why this group exists. And because of that, VDWR manages Smith Mountain Lake as a combined trophy and harvest fishery. So that's why you have your 2 per person per day limit. That's part of that harvest component. And then that protective slot limit during the winter between 30 and 40 inches, that's preserving those trophy fish. And then the no limit during the summer is under the expectation that fish are dying along the lines of those previous studies. And then also VDWR just generally advises those anglers that are contributing or participating in catch and release that you're using those best practices, whether those are rubber nets, not using gloves, supporting fish with both hands as you see in the pictures here, and then avoiding excessive handling where you can. If you're planning to release a fish, you're not passing it to 6, 7 people for pictures before you're putting it back in the water, that sort of thing. So again, what is catch and release mortality here at Smith Mountain Lake? That's been the entire basis of why I'm here, of why I've been doing this research. And so now we can kind of get into what I was actually doing. So what we did was over the past 2 years, between 2024 and 2025, I caught and tagged and released 124 striped bass here in Smith Mountain Lake. This figure on the right is showing the map of the locations where I caught those fish, where the darker circles are just indicating that there more than one fish was caught at that given location. The pictures on the left here, those are just drawings of the example baits that I used, which has all been stuff that you guys have recommended to me. So whether those are hit sticks, F-18s, your Cast King spoons, jig heads, or circle hooks on Carolina rigs for using live bait. Every time that we caught one of those fish, we're recording the total length, the fight time from when we was first hooked up to when it's in the boat, the handling time, how long it's been from being netted, getting that hook out, tagging it, and putting it back in the water. Hooking depth, surface temperature, bait type, and then any post-release behavior, what it's doing after we've put it back in the water. Oh, we're beating each other to it. And this is just an example of those tags that we used and how we put those into those fish. And so I'm just gonna go right into the results since we don't have a lot of time tonight. What we're looking at here, these are box plots of whether fish that survived versus fish that died, and in this case, it's their total length. The wings that we see on these graphs are showing the density of observations. So in this case, we have more fish caught that were 26 inches that lived than fish that were caught that were 22 inches or 28, 30 inches. So that's just kind of what we're dealing with here. And when we go and we do our statistical analyses for length, we found no significant differences between fish that survived or died based on their length. Same thing applies for fight time. What we'll see in these box plots, those dark circles are outliers. Specifically for fight time, we wanted to see if playing with a fish on the line for longer would actually cause that fish to die. And so all these dots are from us manipulating fight time. There's a lot of dots on the blue side of this graph. So we could not kill fish by trying to hold them on the line, meaning fight time really wasn't making a difference here. And our statistical analyses reflected that. Handling time, same thing. No statistical differences there. And that's kind of like a nice thing for me because that means regardless of whether that fish lived or died, we're giving it the same treatment as far as removing hooks, tagging it, and things like that. So for this is a good thing to see for us. Depth hooked. Again, no significant differences there. Now, a lot of you are probably looking at, well, this wing's a little bit wider here than here. So within the data, there are more dead fish that were caught shallower than, at least within that sample. But remember, we only have 28 fish that died within our total sample. So what that means is, like, the distribution just doesn't reflect the variability that we saw with all the fish that we caught. So yeah, there's more within that group, but across the groups, there's no statistical differences. Surface temperature, same deal. Yeah, it's kind of the same thing. There's no differences in surface temperature. Whatever temperature it was, we were catching fish at the same time, at the same time of year, and they were dying basically at random. All right, so this is going to be a little bit different. So we have bar plots with the proportion of fish that died in red, and you have the percentages here in black, versus blue would be the ones that live, same color scheme. The only main thing here is these red dashed lines. Those are actually the estimated catch-release mortality estimates from those previous studies for Smith Mountain Lake temperatures. What we can see here is that for both artificial baits and live baits, our mortality estimates were lower than what was previously estimated. And more strikingly is how different our estimates for live bait was compared to previous estimates. And what this comes down to is those previous studies were in the late 90s, early 2000s, and so all of their live bait was conducted using J-hooks. This is well before the pretty established philosophy that circle hooks reduce catch and release mortality. And so since most of the people that I know that fish here are using circle hooks in general, we were using that for our study, and we're seeing that that is reducing catch and release mortality compared to those historic estimates. Now, but when we look at the groups between each other for our study, there still isn't any statistical differences between using an artificial lure or live bait here at Smith Mountain Lake with these techniques. If we expand that to specific lures, where here circle is talking about those circle hooks again, multi-treble is talking about jerk baits, your F-18s, your hit sticks. Single is referencing single hook for jigs, so those single J hooks. And then single treble is talking about spoons. And between these groups, again, there's no statistical differences here. We do see that there's 35% for the multi-treble hooks, but again, I have to reference that we have, it's really hard to see for you guys, but this is 17 fish that were caught with jerk baits, and that's not a big sample size to be able to make those kinds of connections. So, but I mean, if we had unlimited funds, if we can catch all the fish we wanted, maybe that would be the same, but as far as we can see right now, there are no differences between gear types. All right, but we did find a statistical difference. I know probably people are thinking that, oh, there's not anything happening, but we did find one. But unsurprisingly, it's that for when you catch and release a striped bass, and you put that fish back in the water, if it was floating for any amount of time before swimming down, it has a 52% chance of mortality. So that's not surprising. It's not doing well. It's probably going to die. That was a significant difference. And really what the main advice that we have for that is, you know, if you are doing catch and release during the summer and you're seeing a fish not doing too well, probably if you have the room in your bag limit, that would be a good thing to just, you know, why don't you just harvest that fish and hold on to it, because odds are it's not going to do very well. But with that, that's about the most advice we can give in terms of that. But overall, catch-release mortality during the summer at Smith Mountain Lake is 24%. So if you're going and you're participating in catch and release during the summer, 4 out of the 4 fish that you release, 1 of them will die. We cannot predict why that is. It's just coming down to fish are individuals and they have individual tolerances to stress. So some fish will do better and some fish will do worse. But 24% is kind of the magic number for the 2 years of work that I've been doing here on this reservoir. So now that we know, okay, 24%, how do we use this information to understand the effects that catch and release mortality has on the growth potential of striped bass? How does it affect these fish reaching the lengths that they're achieving? So what is growth potential? growth potential for this case is what is the proportion of fish that are reaching 30 inches or more. And so how do we think about what makes a fish grow? Well, it comes down to longevity, how long that fish is alive for, as well as growing conditions. What are the things that are going into that fish that are allowing it to get to the size that it's getting? Longevity comes down to what are the things that are killing these fish. That could be, those sources are natural mortality, catch and release mortality like I was just talking about, as well as harvest. Growing conditions, that comes down to food availability, whether that's the food, the numbers of bait that's in the reservoir, or from the food available per striper from competition. And we kind of did some number crunching on that, where we looked at the numbers stocked and how that relates to growth. And essentially there is a decreasing trend where with the more fish stocked, you lose the length by age 2. And so essentially the way it works down to is compared to like 250,000 fingerlings, a 500,000 fingerling stocking could reduce length by about an inch on average. So what that comes down to is that there are consequences to growth when everyone's fighting for the same resources. Other things that go into growth are temperature. Fish are cold-blooded organisms. That means that their body, their metabolism, is regulated by temperature. So when temperature is warmer, those fish will be able to grow faster. Temperature is cooler, they're going to be growing slower. And the last thing is genetics. I know it's probably hard to see in this corner, but genetics. Some fish are just better growers than others, and that's just how the population works out. So, and just another visual to think about this is kind of the similar age on the x-axis, length on the y-axis that Dan was showing earlier. All these purple dots is literally every fish that has been aged by Dan or the project from you guys have been turning it in or from people previously. And really what we want to know is what is the fraction of these purple dots that are making it to this green box, which is 30 inches or better, regardless of what age they're making it. So how can we simulate this? What we did was we took a theoretical population of 1,000 age 3 striped bass. We chose age 3 because that's when those fish are fully recruited to the fishery. You know, you can sometimes catch a 2-year-old, sometimes catch a 1-year-old, but that's kind of a fluke thing. By the time that 3 years old, they have an equal probability of being caught by anglers across that year class. Then what we did is we let them grow, and we used that based off the data from fish heads as well as gill nets. So all that growth data, we used that to grow these out to the next age, in this case, age 4. And then we had to kill some of them. And so we did that by the total annual mortality rate, which we were able to estimate by that tag return study. That's why that was so important, because it gave us the proportion of fish in the population that are harvested last year. Those are the portion of fish that were released last year. We then combined that with my catch and release mortality estimate, so then we could get what is the proportion of fish that are dying from catch and release. And then to get that natural mortality estimate, we borrowed from a Baden Lake study, so then we could get the whole picture of how many fish are dying each year. Then we do that for each age going out until either we get to a fish reaching the age of 22, which is the oldest observed striped bass ever recorded in Smith Mountain Lake, or that whole population died. We do that 6,000 more times for each of our simulations, of which we had 8 simulations to go through. And so this graph is basically the culmination of everything that I've done for Smith Mountain Lake, where this red box is the simulation for the current estimates of catch and release exploitation, how many fish are being caught and released during the summer, and catch and release mortality, how many fish are dying from catch and release during the summer. Under those estimates, 20% of the population of age 3 striped bass are making it to 30 inches or more. We also let those estimates vary by plus or minus 10% on my catch and release mortality estimates. When we do that on the same estimates of catch and release exploitation, we have less than 1% difference on either end of those estimates. So what's getting there, under the current rate of catch and release happening at Smith Mountain Lake, even if there is variation in how many are dying from catch and release, the population is pretty robust to those changes. But we also wanted to look at, well, what happens if we do a scenario where, I don't know, maybe we have a regulation that says we're not going to do any catch and release during the summer because we want to just keep all the fish alive, we want to do all that stuff, right? So that is this scenario right here. The improvement from the current estimates is 1.3%. So not really anything that we could notice in terms of fish 30 inches or more. We went and did some more estimates of, okay, well, what if exploitation is higher during the summer by 2.5 times, just to throw a number out there? Well, under the same levels of catch and release mortality that we estimated before, there are decreases in the proportion of fish that make it to 30 inches, with the biggest decrease being about 4%. So for scale, that would be if there was 10% exploitation. So that means 10% of the population of Smith Mountain Lake is being fished just between June and August, and catch release mortality is 34%, then we could see a 20% decrease in fish that are 30 inches or more. We also did a very extreme scenario just to see how low the potential could be to reduce striped bass length potential. And when we multiply our exploitation estimate by 5 times, that means for every trip that you do and you're doing only catch and release, you have to multiply 5 boats to go out with you. We could see losses in those proportion of fish that make it to 30 inches by almost 8%. And so that's nearly half. So what we see is there are potential risks for catch and release mortality affecting the length potential. But in order to make that happen, we have to have extreme scenarios of catch and release exploitation, meaning the amount of people actively fishing for these fish every day has to be substantially more than what we're currently seeing. So just the last couple take-home points, so before we get into questions. Catch and release was lower than what was previously estimated for Smith Mountain Lake. Particularly, live bait, for live bait, using circle hooks works, and particularly using inline circle hooks. We used a mix of straight eye or octopus eye inline circle hooks for that. Those differences can be up to you guys, but specifically inline circle hooks were what we were using, and if you're using those gears, I would estimate that you should expect those kinds of levels of catch and release mortality. Under the current scenario of catch and release exploitation and catch and release mortality, the effects of catch and release mortality on length potential are negligible, but there could be potential for differences if exploitation reaches unattainable levels. Angler participation and collaboration has been critical to the success of my thesis as well as the efforts by Dan. Without you guys in and out every day, submitting fish heads, helping with gill nets, turning in reward tags that pay for your gas, helping me out, whether teaching me where I need to be, calling me up saying, hey, there's fish over here so I can tag some of these fish. All this stuff has been crucial for me to be able to complete this thesis and be able to give you guys the best science that we can give you. So thank you. And lastly, I know probably a lot of you are thinking, well, what about those citation fish, those 37-inch plus? And unfortunately, just the reality of it, the data isn't there to be able to make comments on that. And what it comes down to is not just being able to catch and release those fish with tags, but for fish heads particularly. We don't have enough on the growth of those fish to really understand how frequent those are, because what we know is there are citations submitted and given out to anglers over the course of the year, and that's it could be like 8 or 9 or so in a year, but we only have 15 fish over 40 inches in the whole data set for the past 25 years. So if you think about it that way, there's not a lot of data for those fish that are those jumbos. But with you guys' help in submitting those fish heads for us to be able to analyze, we can actually go back later and update these models to be able to give you guys some information on that. And just real quick, I just want to thank all my contributors, to everyone that was here and helped me out, between the technicians that were fishing with me for the past 8 months out of the 2 years I've been here, or 4 months, sorry. The Virginia Department of Wildlife biologists, George Palmer and Tyler Young, that are helping me intermittently. The Federal Aid and Sport Fish Restoration Project for funding this research. Jeff Smith in particular for showing me how to fish for striper here in Smith Mountain Lake. And you guys, your support has been awesome in being able to, for me to improve as a scientist and as an angler, and particularly to John Anderson and John Hughes for housing me and my crew for various times, as well as Tim Barr for supplying the SML Plus water temperature data. And then just a special thanks to the friends and family that came out and were able to support me in this project. And with that, I will take any questions. Yes, sir.
How many fish did you catch for the 2 years you were here total?
124 were tagged. We caught maybe 60 more than that, so almost 200 total.
What was some of the farthest travels that tagged fish would go?
Of the ones that off the top of my head, I think about...
Wait a minute, Nathan, you don't have to answer that. John Hughes is just, he's looking for information.
For the ones on, well, within a 2 week period, I think 8 kilometers is what's coming off the top of my head. So like from Betty and Becky's to, I don't know, it's like R31 or something, or past it. So pretty far.
But these were done in the summertime when the upper ends of the lake, for the most part, were warm and the fish were staying in the lower parts of the lake. And he didn't track them for a long time. He tracked them for a few days or so until to make sure that they were still, there was a short period of time to make sure that fish was living. Once it was, makes it through 4 or 5 days, that fish is not going to die from catch and release mortality. How long did it take the fish to die after they were caught? Within a day?
Yeah, so when we were tagging and tracking these fish, we wanted to look at that 72-hour time scale so we could see what is catch and release mortality. If we look beyond that and we're trying to see if fish died, weeks and weeks and weeks later, then we can't attribute that to the angling event. It could be other things that would be causing that mortality. John?
So are you saying that the catching of these practices that are going on now are as good as they can be, or do you have any specific suggestions for leads in one direction or the other?
That's going to come down to what, as far as what are people actively doing on the lake. As often I was out there and fishing with many of you, I'm not policing, so I don't know what actual activities you guys are doing on a given day. But for the best practices that we listed earlier, if you're doing those kinds of things, if it gets to it.
Oh, so circle hooks.
Wet hands, rubber nets.
Yeah. Rubber knotless nets, that's a big thing. You're not scraping up the fish. Basically, things that you're keeping that slime coat on. Gloves, nets with knots that can scrape the slime coat off, and that can cause issues.
So, in other words, if you're going to do catch and release, do it the right way.
Yeah, right. Like if you're thinking about doing catch and release, you want to have that with your first thought in mind. Okay. You don't want to be harvesting and then fishing in a certain way that if you think there would be issues, then you want to be thinking about that ahead of time before you're going out. But like if you're going to be using live bait, using those inline circle hooks, you're going to be pretty set for whether harvesting or catch and release.
So I just want to clarify something. We're looking at the no length limit when the water's warm. Okay, so you really don't want to release when the water's warm, correct?
That's kind of the under the press. That's under the.
Before we did this, the data that was out there from other lakes that weren't quite like this said you're probably killing 80+% of the fish if you throw them back in the heat of the summer.
I'll agree with
So,
you. Now, when the water's colder, and you want to release you want to release them between 30 and 40, how about if you catch something lower than that? I mean, what's the limit there? Is it still the 20 or 22 inches that it used to be? That you want to keep anything under that, correct? In the summer, yes.
there is no size limit in the summer. Any you can keep any size you want, whether it's 6 inches or 40 inches, and then in that fall period, it has to be basically under 30 inches.
Right, okay, but between November 1st and May 31st, we know you want to let the big ones go because you want the big ones. But what about the lower end?
Those could be harvested and released during the winter
But
time.
is there, you don't want the young ones, I mean, so where are we at, 20, 22 inches? It's got to be at least that?
No, it can be 12 inches, any size.
Okay.
Any size less than 30 is open.
The point during the winter is you want to extend the life of fish that are between 30 and 40 inches. Those are what we're considering those trophy fish.
Because they survive really
So thinking
well
of it,
being
you're
caught.
saving the big ones, and then those under that, those are free for harvest.
Okay. I was always under the impression there was a length limit, even in
No,
It's not even-
no minimum, no, nothing down there.
Okay. So I asked Chad this, but Dan, do we know what we're doing this year as far as?
Did I just breeze right over that?
You did.
It was one of those little bars on there.
Way back there.
I forgot to mention it. There was one of those little bars. My plan this year is 325.
325, okay. My wife's been asking it, so I didn't know that.
Okay. Shoot.
Nathan, in your releasing of the fish during the summer, did you vary your release technique as far as the survival?
No, and really the point of that was, and there's a whole part of catch and release mortality studies of how do you release a fish, and so one of those aspects is what's called a ramp score, and so how that works is basically they're looking at different indications of stress that could potentially predict mortality. And so there's basically different senses that they're checking off that are either impaired or not. But how you do that is you're usually holding that fish in the water and you're testing the reactions to those senses. With us doing a summer catch and release mortality study, knowing that the surface water is inherently stressful to those fish, we didn't do that in the hopes of, we don't want to add excessive stress from our procedures that would be counterintuitive to what anglers are actually doing. And so for all of our releases, I know a lot of you have heard of the torpedo release, whatever you want to call it, we're just putting them back in and letting those fish handle them, sort themselves out. And so, fish is just going in the water. We're not going to keep it holding up next to the boat, stressing out even longer. And whether those fish were floating for a little bit or they just swam off, that's where we were getting those releases.
I asked them when they started this to basically do the best they can to give that fish the best opportunity to survive. Do things as quick and clean, as fast as they possibly could, to know what survival would be under best case scenario if people did all the right things and tried to do it as fast and as thorough as possible. That's what I asked them to do, and then after the first summer, they mixed it up a little bit like the fight time. A few fish, they just took longer to reel in. I mean, the whole first year it was hook a fish and try and horse it in the boat, get it in as quick as possible, not let it build up too much lactic acid, not take too long. But most of his study was trying to concentrate on giving the fish the best chance of survival. If we could do this for 10 years, it would be a great opportunity to branch out. Okay, now we know the baseline. Well, let's look at this part. And the problem is if you only do it with a few fish, you really can't say a whole lot. You have to have quite a few fish in a sample of doing something different to really say that it made a difference or not. So he didn't have enough time and enough fish that really varied that stuff up. But the whole intent was for folks to be aware that these fish do need a little special care in the summer and give you data from or what's going to happen under the best case scenario, handle them as quickly and as safely as possible.
Yes, sir.
I was wondering the difference between Leesville and Smith as opposed to the growth of the fish and how well they survive.
And that would be a fantastic question for Dan, because I only did Smith Mountain Lake.
What was the question?
The growth between Leesville and Smith Mountain.
Why are they growing better there?
Yeah.
They're eating food. Our numbers are much lower, a lot less stripers there, and they have a whole lot more food, so they maximize their growth. That's what they're the same fish, come from the same hatchery, hauled in the same trucks, stocked out of the same boat. There's nothing different about them. It's just that we put them in there and the densities are much lower, the food's a lot better, and they're growing to their potential. That's the way they should be growing if they eat what they want throughout their life.
So we should be fishing more in Leesville.
There's some people down there that would beg to differ.
There was no difference in mortality from the pet fish club fishing?
No, and that's surprising.
Air bladder?
Right, so the question is like why, like we didn't see any differences with depth. And so for a lot of marine species like grouper and snapper, those kinds of species, they typically have problems with being caught at depth, bringing it up, and their gas bladder inflates and they're not able to regulate their buoyancy. So yeah, it's an interesting question. And we fished all sorts of depths, and we pulled a lot of these fish out from 80 to 100 feet. That's 3 times the atmospheric pressure at the surface. So unless they have some biological adaptation for adapting their buoyancy rather quickly, that would explain why we weren't seeing any really barotrauma associated with mortality. And the other thing I would add to that is if we're going and we're looking at a graph of fish, like on your hummingbird or whatever, a lot of times during the summer, you're going to see fish going from 90 up to 30 and then back down to 90 in a couple seconds. Like they're actively swimming and making these big depth changes on their own. So I think from those observations that the rapid ascent isn't contributing to any barotrauma because they're already doing those kinds of movements for like daily, repeatedly, all the time. So I think they just have some adaptation to barotrauma. That's why we weren't seeing that.
They're built for it. That was one thing I was pretty shocked about. I really thought if you're catching a fish 100 and 110 foot of water, that thing is going to be in bad shape. You put it in the boat, and it didn't make any difference.
So, will you be around after the meeting?
Yeah. Any more questions? I'm going to be hanging out.
Time we got to wrap up, but I just want to say thank you very much.
Thank you. Thank you, guys.
The growth